Tumors need a blood supply for growth, which is achieved by inducing angiogenesis or exploiting existing blood vessels. The primary mechanism in tumors is sprouting angiogenesis, where new vessels form through the growth of endothelial cells. A less common method is interceptive angiogenesis, which involves a splitting of the existing vessels and operating independently of growth factor, so making it more resistant to some anti-angiogenic therapies. The rapid angiogenesis also resulting in a leaky vasculature with gaps between endothelial cells, which these gap size between endothelial cells, also called interendothelial gaps, could be up to 2000 nanometer. These gaps, alongside with other characteristics of the tumor, allow macromolecules and nanoparticles to penetrate the tumor tissue more effective than the normal tissues. This effect is called, as we talked before, EPR effect, which was discovered by Professor Hiroshi Maeda in 1986. He already continued to understand the overlooked factor in EPR effect, and he was an editor for a special issue of EPR effect-based tumor-targeted nanomedicine, which was published after his death by MDPI. Most of the examples of failed cases reflect the use of so-called nanomedicines with very poor plasma half-lives in vivo or active pharmaceutical ingredients in nanomedicine that rapidly became free low molecular weight drugs. So this text itself indicates that there is an issue for translating EPR effect to the clinics. So the main question is, which characteristics of the tumor enhances the EPR effect? As we talked, the first is the massive irregular vascularization of the tumor, which has made a leaky structure, facilitating the macromolecular and nanoparticle arrival to the tumor. The other, in addition, is a huge amount of VEGF and biochemical compounds such as nitric oxide, bradykinin, etc., which provide a favorable condition for EPR. And for example, we know that the nitric oxide increases the blood flow, and therefore this overwhelming of such biochemical materials will increase the retention of the nanoparticle inside the tumor. The other factor, which is very important, is the lack of lymphatic drainage in tumors, which extends the presence of the macromolecular drugs in the tumor structure. This selective accumulation enhances the efficacy of nanoparticle-based drug delivery system, improving the therapeutic index by concentrating the drug at the tumor and minimizing the systemic toxicity. So, the main question: what is the main problem of EPR in clinics? The first one is the missed clinical comparison of nanodrug with free drug. It is well known that the protein larger than albumin, which is 48 kilodalton, and macromolecules larger than 40 kilodalton leak out from the tumor vessels and accumulate in tumor tissues. As a prerequisite of EPR effect lower than this size range, the drug will not remain in blood plasma for at least 6 hours. The other factor which determines the circulation time is their interaction with immune system and nanoparticles. Characteristics such as charge, hydrophobicity, and morphology, etc. is associated with this parameter. I will subdivide this part in two main issues. The missed clinical comparison of nanodrug with free drug. The first is that in drugs with plasma protein binding, the EPR effect drives the tumor targeting also. However, in most nanoparticle studies in clinical trials or preclinical studies, most of them are restricted to the comparison of the nanoparticle in the tumor and the adjacent healthy tissues. However, the comparison of nanoparticle with drug should be assessed because some drugs has some protein binding and they also are benefiting from EPR effect, and this effect is seen in the drugs also. And the other thing is accumulation of nanoparticle is not only because of EPR, but might be due to preferential homing. It was a study by Northfeld in 1996, and it was shown that the doxil to doxorubicin in ARKS lesions and found that doxil levels were 5.2 to 11.4 times greater than those of doxorubicin. But the key point here is that the liposomal doxorubicin tends to be accumulated in dermal and epidermal tissue, and in some cases a disease called hand-foot syndrome was diagnosed using liposomal doxorubicin. Considering that ARK ERKS also arises from the spindle cells and the dermis tissue of the skin. It is difficult to conclude whether the increase of drug concentration in ERKS lesions is related to the EPR effect or preferred accumulation of doxil in the epidermal and dermal tissues compared to the free doxorubicin. And also, it is very interesting to mention that in most cases, this is not the EPR effect. We have some other methods for entering the nanoparticles inside the tumors. For example, transendothelial enter to the tumors. EPR effect is not the only method which nanoparticles or drugs enter to the tumor. Namely, for example, in a very great study by Chen, they initially counted the number of the gaps in the 313 vasculature of U85 mg glioblastoma xenograft using TEM, and they found 500 gaps per millimeter square in vasculature, which is about 0.048% of the blood vessel surface area. Next, they did simulation to see how much gold nanoparticle should be found in the tumor using EPR mathematical models, and they found that about 0.0158% injected dose per gram should be in the tissue. However, the ICP-MS results of the tumor for intravenously administered gold nanoparticles showed a higher number, which was 0.63% injected dose per gram. It means that for such amount of AU nanoparticles in the tumor, it must be about 30,000 gaps per millimeter square, which is 60-fold more gaps versus the real, which was 500. So these data shows that in overall, 97% of gold nanoparticles in U87 mg glioblastoma xenograft models enters through the active processes, and only 3% were using the EPR effects. The other factor is that the most xenografts are showing a higher EPR effect versus human tumors. Most studies in nanomedicine are using the xenograft models. However, the xenograft models are highly vascularized and have fast proliferation rate, high level of macrophage infiltration, and therefore they are mostly showing an exaggerated delivery efficacy. And therefore, this is unnecessary to develop a transgenic models or patient-derived tumor explants, which resembles more the human tumor. And in the other words, the preclinical studies must be some models which pathophysiologically can resemble more the tumor's structure. To clarify the questions on nanomedicine design based on EPR effect, Luan et al. Tested if commercialized nanomedicines such as Doxil, Myocat, Abraxane, Genoxel, PM, and other brands could enhance tumor accumulation by EPR effect versus normal breast tissue in 3 different breast cancer mouse models, which were transgenic mouse spontaneous, orthotopic, and subcutaneous breast cancer. They initially analyzed tumor accumulation of small molecules, which was Evans Blue and doxorubicin, in tumors versus the normal breast tissues in 3 different mouse breast cancer models that we talked, with the same cancer type and same genetic background. When breast tumors were compared with the normal breast tissues, nanomedicines, drug solution, and clinical micelle formulation enhanced the tumor accumulation in all 3 different breast cancer models. So there were no difference when you compare the free drug and the nanomedicine. However, when nanomedicines were compared with drug solution or clinical micelle formulation in tumors, nanomedicine did not enhance tumor accumulation in transgenic mouse spontaneous breast cancer, although they did in subcutaneous and orthotopic breast cancer. So this data also shows that a difference between the models that we use is very important, and also in some cases when you compare the free drug and the nanoparticle, the results would be so close and there is no high margin difference. The other factor is EPR heterogeneity. We know that the tumors are highly heterogeneous and vary by the size, for example, from 1 millimeter to several centimeter, or they could be metastatic or primary tumor. They could have different treatment of history, or for example, the vascular density regarding to the tumor type could maybe change. And moreover, the vascular structure is dynamic and changes over different stages. And therefore, we have a definition called EPR heterogeneity. Heterogeneity is the reason, this kind of heterogeneity is the reason that white doxil is not entering well to the solid tumors, such as breast cancer, but works, for example, in ARKS.